Hello, everyone. Welcome back to Genetics. It is time for translation. This is where we go from RNA to protein. And today, for the first time this semester, we will be leaving the nucleus for parts unknown. Well, actually for the cytoplasm, but there you go. What are we learning about today? Well, a handful of things. But we're going to start off with how do you translate? Like I mentioned last lecture that the difference between transcription and translation was with transcription you were writing down a copy of something. So you're going from DNA to RNA, you're in the same language, you're just writing it on a copy. But for translation, you're going from one language to another. What's our translator? Who's our translator? How do we know what the other language is and how to get from one language to another? You can't throw DNA nucleotide sequences into Google Translate and get protein out. It would be cool if you could, though. So, how do we know what the amino acid sequences are going to be based on a nucleotide sequence? This was worked out in the early 1960s. There were a lot of ideas. After the double helix structure was identified in the early 1950s, a lot of people then went, okay, so that's going to be how the genetic code is, stored and kept. And sure, we can see how the hydrogen bonds of the double helix can break pretty easily, and that's going to, form template strands for easy copying. And sure, we can see how that would be a great way to go from storing information to, duplicating it for synthesis. But how do you go from that to protein? Well, this is before mRNA was discovered. When people were trying to figure out how to go from a DNA sequence to a protein. And I shouldn't say this is before mRNA is discovered. This is about the same time that mRNA was discovered. I'm going to throw a handful of names at you right now, or over the next few slides. It's not terribly important that you remember them, but some of these are names that have come up already in past lectures. So a guy by the name of George Gamow, he's been hanging around the periphery of most of the story so far this semester, but he's never been on stage and participated in anything. George Gamow was the first person to say, hey, I bet every 3 nucleotides as a triplet encodes 1 amino acid. And his reasoning, his rationale, and you may have heard all of this before, his rationale was you have 20 amino acids that are going to be in any given protein, and you've only got 4 nucleotides. So if there's 1 nucleotide encoding the amino acid, well, then you can only encode for 4 amino acids, adenine might encode for methionine, and cysteine might code for phenylalanine, and guanine might code for lysine, and if you have 2 nucleotides encoding an amino acid, well, that's 4 squared, because 4 possible nucleotides in the first position, 4 possible nucleotides in the second position, so 4 times 4 is 16. Well, that's still not enough to cover all 20 amino acids. AA might be leucine, AC might be threonine, AG might be phenylalanine, that kind of thing. But that only will get you to 16 amino acids. But the problem was, once you went to 3 nucleotides, well, that's 4 times 4 times 4. And that's 64. And then the question becomes, there's only 20 amino acids. Why would you have a code that allows for 64 possibilities if there's only 20 amino acids? It can't be right. But there's nothing else it could be. So there's a lot of confusion. And there was even the suggestion that, well, maybe some of those 64 possibilities just don't exist in DNA sequence. But, lo and behold, every possible combination of nucleotides is present in DNA sequence. So what were some other possibilities? Well, one possibility was maybe it was a 3 or 4 base code, but one of the bases was a separator between codons. So in this case, if you're not familiar with the word, codon is the number of the set of nucleotides that encodes one amino acid. Okay, so at this point in history, we all know that 3 nucleotides forms one codon, and that encodes for one amino acid. But they didn't know this yet. And so they thought, well, how does the machinery of the cell know which bases to consider a codon? Maybe there's word separators between them. The nucleotides encoding a amino acid, and then some pattern that says that codon ended, and now a new codon is about to begin. And so this was considered the comma hypothesis. Like there was a codon, comma, codon, comma. And then the next question was, okay, no matter how many nucleotides there are in a codon, is it an overlapping code or is it a non-overlapping code? And the image on the screen here shows the differences. With a non-overlapping code, you have one codon, and then you move on just past the end of that one, and then you start the next codon, and you move just past the end of that one and start the third codon. Whereas an overlapping sequence is starting from base 1, however many bases long it is, that's the codon. And then you start from base 2, and however many bases it is, that's the codon, and so on. And so here's where we get back to some familiar names. Francis Crick and Sidney Brenner. So Francis Crick and Jim Watson worked together to discover the double helix. Crick was an Englishman working at Cambridge in England, and Jim Watson was an American from Chicago who had come over to do some graduate work in England. And when he finished up and he graduated, he left, and at that point he already had discovered the double helix structure, and so he got a job at Harvard right out of the gate. But when he left, he and Francis Crick at some point had been sharing an office, and when he left, Crick had an opening in his office, and there was a South African man, Sidney Brenner, incredible life story, illiterate parents. His father was a shoemaker. He grew up learning how to read by teaching himself on the newspapers in his father's shoe shop. And then one particular customer saw him trying to teach himself how to read and became a benefactor and basically sent him to private schools and gave him tutors. And then he wound up going off to medical school at 14 years old, and then left medical school before the end to go to graduate school and get a PhD. And then he wound up in Cambridge. Or actually not in Cambridge, but in London. And he kept visiting and hanging out in Cambridge. So he was one of the first people to see the double helix before it was widely distributed. So Sidney Brenner had struck up a friendship with Francis Crick, and Crick basically talked to his supervisors and said, we need to give this guy a job. I mean, he is brilliant. So they said, okay, well, you're going to watch over him. So they put Sidney Brenner in Jim Watson's spot in Crick's office. And for the next 40 years, Brenner and Crick shared an office together. And they spent their days just discussing logic. The logic of how a cell could possibly work, and how to figure out if your idea is right or wrong. And so they would come up with a question. First they would look at a problem in science. Okay, what do we need to understand? And then they would boil that down to a specifically worded question and say, okay, the way to get it, that thing we're trying to understand, is we need to answer this question. And these are the experiments that we could do that will answer that question. And here's all the possible results we might get from those experiments. And then, based on what the results we got are, these are the possible answers to the question. And it will either confirm or reject or alter our hypothesis. That's real hypothesis-driven science, and that's all they did all day long for 40 years. Half of their papers are theoretical. Well, I shouldn't say that. I should say they laid out theory of what they were going to do for the first half of their paper and then confirmed it with like 2 or 3 experiments in each one. And considering the way science is done today, it's just mind-blowing and brilliant to see their work. And so they decided to take on the concept of the genetic code, how nucleotides could encode for amino acids in proteins. And they took George Gamow idea that it was possibly a triplet code. And, they agreed it couldn't be a single base or two bases, but it could be 3, 4, 5, however many, who knows? It could be variable, who knows? And so they set up an experiment to prove, or to answer the question of whether or not the code is overlapping, and to answer the question of how many nucleotides, and to answer the question of are there commas. So before we get to their experiment, let's just think about the results of, or not the results, but let's say the end product or the effects of mutations in DNA given an overlapping code or a non-overlapping code, or a comma or no comma. So I've got here left and right overlapping and non-overlapping codes, and the little brackets underneath indicate what the 3 base codons are. And for now, we're just going to assume it's 3 bases because we know we're right. The numbers underneath each are, I'm just throwing random numbers saying that ABC is a codon that encodes amino acid 1. BCD encodes amino acid 2. CDE, 3, and so on. So for this sequence of ABCDE, 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 you can tell pretty easily our amino acid sequence, if we have an overlapping code, is 1, 2, 3, 4, 5, repeating. However, if we have a non-overlapping code, and again, we're assuming it's 3 nucleotides long for each codon, then the amino acid sequence we have is much shorter. And it is not the 1, 2, 3, 4, 5 repeating, it is 1, 4, 2, 5, 3. Now, this would also be a repeating sequence, but there you go. So that's going to be the difference in protein sequence based on the differences in nucleotide sequence. You follow? Now, what happens if we delete a base? If we delete that very first B, with an overlapping code, we are only going to see an effect in the first 2 amino acids. The effect we're going to see is the very first codon should have been ABC, but now it is ACD. So I'm just going to call that amino acid number 8 because it's different. The second codon should have been BCD, but without the B, it shifts, and now it's CDE, and that's just the codon giving us amino acid 3. And so the entire rest of the sequence, if you look at it, is exactly the same. 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, and so on. So the only effects are going to be right in the immediate vicinity of that deletion. Just the codons that deleted base were in. If you look at the non-overlapping code, if you delete a base early on, every amino acid following it gets changed. If you delete a base late, like if we deleted the last B instead of the first B, then really only the last couple of only the last couple of amino acids in this would get changed. So here's your difference, and this is the important thing to remember. The difference is if you have an overlapping code, a deletion of a base, or if you add in an extra base called an insertion, that is only going to change the amino acids right where that base change is. With a non-overlapping code, if you remove or insert a base, all of the amino acids following that codon are going to be changed. That's a huge difference in effects. Now, whether there's commas or no commas, one way to think of that would be, just a random nucleotide or multitude of nucleotides, whatever multiplication factor, in between each codon is meaningless or has a different meaning. Whereas no commas is just each nucleotide, each codon, one after another, is back to back to back. So these were the possibilities that Crick and Brenner had in front of them. And they tried to figure out the best way to determine what was going on. And they realized that someone else in a different lab had been doing a set of experiments where they were treating nucleotides, or rather, they were treating the cells of, I think it was either bacteria or yeast, I can't remember what, with specific dyes. And they found that the dyes were changing the size of the chromosomes very minutely. And the amount of change depended on what concentration of dye they added, and eventually they were able to prove that specific dyes added or caused the addition of nucleotides into the chromosome, and certain dyes caused the removal of nucleotides. Or rather, this one particular dye could either add or remove nucleotides. But it did it one base at a time. So they treated, let's say it was bacteria, with this particular dye, and it wound up intercalating into the DNA and caused the DNA to either, at the next replication, insert a base or remove a base, but only one base at a time. And that was all Crick and Brenner needed. So this is a figure from their paper, and I don't expect you to understand it because it's not an easily understood figure. But the take-home message was they found out that they could classify. They treated a bunch of bacteria with these dyes at different concentrations, and they found that if there was the addition or removal of a single base, then there was a massive change in the function of the protein. And basically, specific amino acids in the protein were lost, changed, removed, whatever. And they could map where the changes were based on homologous recombination. So they had different strains of the bacteria, and they treated one, and then they allowed for homologous recombination, and they could find, based on that, where the changes were that caused these mutations. And when they had one base added or removed, or two bases added or removed, it completely messed up and destroyed the protein sequence. But if they had three bases added, or three bases removed, or any multiple of three added or removed, then there was a very small localized change in the amino acid sequence, but the entire protein was essentially functional. And from this, they said, okay, a codon has to be three or a multiple of three nucleotides long. And it has to be a non-overlapping code. And then they used just sheer logic to say the whole concept of a comma has been disproven already by these other experiments that other people have already published. It's just no one thought to look at each of those three different or four different experiments side by side. But when you combine those experiments side by side, the results suggest and prove there can't be a comma. We don't even have to do the experiment. It's already been done. So this paper came out in 1961 in Nature, and honest to goodness, it should have gotten them a Nobel Prize. I mean, Crick already had one, Brenner did not. And this was the first time that he was stolen, first time that he was denied a justified Nobel Prize. It happens again in about 3 years, I think, after this. So based on that, there are 3 different types of mutations you can have in DNA. The first 2 actually have to do with how tRNA works. So we're going to get into that later in this lecture. But the third one, the frame shift, that is very elegantly displayed here. The frame shift is the result of adding or deleting a single base. In this example, you're adding an A at the position 7. And that shifts all of the nucleotides down 1, or 1 to the right. And that changes what amino acids come out. So the ALA that keeps getting repeated on the top is the abbreviation for the amino acid alanine. And the nucleotide sequence under it, GCU, 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 that is a codon that specifies alanine. But when you add an adenine, an A base, into that mRNA, you change the third alanine into a serine. And that is not the only change you make, because now none of the rest of the codons are GCU. Now they've all been shifted by one base, so they're all UGC. And that encodes cysteine. So you can add 2 more bases somewhere in there, and then everything after the third base that is added will be back to alanines. Or you can remove a base anywhere, and everything after it goes back to the correct number of bases, everything after that will be an alanine again. So you have to either add or remove multiples of 3 to retain the majority of the amino acids correct. The region immediately where the changes occur is going to be different, but everything outside of it will be the same. But if you only add or remove 1 or 2 bases, or multiples of 3 plus 1 or 2, or minus 1 or 2, then you are going to mess up everything from there to the end of the amino acid sequence. That is a frame shift mutation because you are shifting what's called the reading frame. The reading frame is literally what is the first base in the codon, second base, third base. Okay, we have now done that. Let's move 3 nucleotides down, and that is our next codon. 3 nucleotides down, that is our next codon. That is the reading frame that you are in. You know what the first base in each codon is, and you move 3 bases down to get to the first base of the next codon. If you shift the reading frame, that means the first base of your codon isn't what it should be. It's now the second or third base of the original codon and not the first. And everything after that is now also shifted. So that is the short version of how DNA as a sequence, as a language, encodes amino acids. 3 bases per codon, no commas, not overlapping. So now let's get into a gene, because these are precursors to what we need to understand for translation. The protein being encoded by the 4 bases as codons, and then this next part, what are the parts of a gene? Okay, so we know that protein is made off of mRNA, and we know that the mRNA is made from DNA as a result of transcription. And we know that in eukaryotes, mRNA consists of a string of RNA nucleotides that's got a 5' guanine cap, and that guanine is attached backwards and methylated. And it has a poly A tail, which is just hundreds of adenines in a row. And that these 2 things keep the RNA from being digested and degraded, at least for a while. Okay, great. That's mRNA. That is the physical structure. But what is a gene? A gene is what the different parts of the RNA do. Now, this can get a little confusing. So I want to take this piecemeal and not from left to right. the part of the mRNA that actually encodes the protein is not the whole mRNA. It is a subset, a section of the mRNA. The part of the RNA that forms protein, that encodes protein, is a subset of the whole RNA. In this case, that is the blue bar that I have labeled gene coding sequence. That is your gene. The mRNA still goes from the 5' cap to the poly A tail. The mRNA is larger than your gene coding sequence. In biological terms, in molecular biology, that is abbreviated CDS, coding sequence. Now, every coding sequence starts with a start codon and it stops with a stop codon. That seems kind of reasonable. The start codon in eukaryotes is always AUG when you're looking at RNA sequence. When you're looking at DNA sequence, of course, it is ATG because the U and the T speak the different languages of DNA and RNA. In prokaryotes, it's usually AUG, but there is some variability allowed. The stop codon, no, hang on, let me reiterate something here. The AUG, that codon, that specifically tells the cellular machinery that there should be an amino acid called methionine at this point. Okay, methionine, it's in every protein, it's all over the place. Each protein could have any number of methionines in them. 20, 30, 170, doesn't matter. Only one of the methionines is considered the start codon. There are exceptions to that, but that's more complicated of a story than we need. So, one specific methionine among many, one specific codon, AUG, encoding a methionine, is your start codon. Usually, not always, but usually, it is the first AUG that comes after the 5' cap. So in this case, that whole purple region there most likely does not have an AUG in it. Now when you have that AUG, that start codon, that defines your reading frame, because that says, here is a codon, this A is base 1 of our reading frame, this U is base 2, and this G is base 3. And so now, you can go to the base after G, and that is going to be base 1 of the next codon, and continue 3 bases down the line, all the way until you reach a stop codon. Now, there are 3 possible stop codons. There's only one start codon, AUG. It can happen multiple different times, but that is the only sequence that forms a start codon in eukaryotes. The stop codon, well, it can happen multiple times in an mRNA, but the very first one that is in the reading frame of the start codon, the very first one is the stop codon. And we'll get into what that actually does in a few minutes. But the sequences of that are TAA, TAU, and TUA. You should know those, actually. I'm not going to make you memorize which codon encodes which amino acid, the genetic code as it is. There's really no need for that because it's easily accessible throughout the entirety of the internet, but some things you just should know. Okay, so the coding sequence, the gene coding sequence, it spans from your start codon to your stop codon. Now, we've got the 5' cap, we've got the poly A tail. What are the purple and the peach regions in this diagram? I've got them labeled as 5' untranslated region and 3' untranslated region. And they are exactly what their name says. They are regions of the gene that do not get translated. And what that means is when the mRNA and the ribosomes get together and form a protein, they ignore the purple and the peach areas. The ribosome does not make protein from the purple and the peach areas. The ribosome shuns the purple and the peach areas. It doesn't care about them. It ignores them. They are not made into protein. They are not like introns where they get transcribed and then cut out later. I mean, these regions do not get translated and then cut out later. No. These don't even get translated to begin with. Okay? They don't get in the car and go to the dance. They're stuck at home. This is a gene. Your 5' cap, your 5' untranslated region, your start codon, your coding sequence, stop codon, 3' untranslated region, and poly A tail. And of course, hopefully you see that the 5' untranslated region is called that because it is to the 5' side of the coding sequence. And the 3' untranslated region is called that because it is to the 3' side of the gene coding sequence. Now, an important abbreviation to understand. Untranslated region is very tedious to say over and over and over again. So very few people actually say that out loud. They are referred to as the 5' UTR and the 3' UTR. U for un, T for translated, R for region. UTR. Together, they are referred to as the UTRs. So each gene has 2 UTRs. One on the 5', one on the 3'. And they do not encode amino acids at all. They can have stop codons willy-nilly any which way you choose. The 3' can have AUGs willy-nilly any which way you choose. The 5' UTR generally does not have AUGs. Okay, so now we've figured out, or we've discussed, how you get from one language to the next in terms of just sheer information encoding. What is our translation software? You know, what's the Duolingo or the Google Translate that gets us from nucleotide to protein? And now we've looked at, on our mRNA, what are we going to use to go from mRNA to protein? Which part of the mRNA is involved in this process? It's not the 5' cap, it's not the poly A tail, and it's neither the UTRs. It's just the coding sequence surrounded by a start and a stop codon. That's all we're dealing with. Okay, so what does the actual work of translation? Ribosomes. Ribosomes do the work. Ribosomes are what else do we need? We need tRNAs. We need some proteins called aminoacyl tRNA synthetase. We need our mRNA, obviously. We need the amino acids. We need some various assorted helper proteins. And we're going to need ATP and GTP. Now, when I was taught genetics, most of this part of the semester was a litany of the names of the different helper proteins. And, you know, this helper protein comes in and does this, then that helper protein comes in and does that. And we had to memorize all the names and which one does which. If you're not going to get into molecular biology as a career, there is no reason on this earth you need to know all their names. However, to understand this process, you need to know what they do. You need to know that there is something doing that job. So I'm not going to bother with, hammering out the names with you. Just like all the various helper proteins and loaders and clamps in transcription, I left out the vast majority of those. They're there, I just didn't really discuss them very much. And the same thing's going to happen here with translation. Trust me when I say, you're welcome. Okay, so what's the process? I know I breezed past the names of all the players, and that's because I don't want to explain them twice. I'm going to be explaining them as we go along. Now, I always tell you that visual learning helps me, and I hope it helps you. Definitely watch one or both of these YouTube links. There's a lot of animations out there about translation, and just like with transcription, a bunch of them I don't think are very useful or helpful, or they're just confusing. These two I liked. The one from Virtual Cell, you can probably skip the first 30 seconds or so. Alright, so we've got our reading frame set. We have a start codon with the AUG, and we have the stop codon with the UAA. And you know what? I misspoke when I said the names of the three stop codons, because I replaced the G's with U instead of replacing the T's with U. So, cross off any notes you might have taken on what the sequences of the stop codons were. They are UAA, UGA, and UAG. Alright, we got that under control? Good. Now, in this diagram, the bases 5' of the start codon are labeled NNN because we really don't care what they are because they're in the 5' UTR. Their purpose is not what we're going to be discussing today. It does have a purpose, but not what we're discussing today. The same is true with the bases after the stop codon. Those ends there because those are the 3' UTR. We're not interested in what they do today. And what they do, though, actually is help control the function of the ribosome. But they don't control the sequence of amino acids that are made or how the amino acids are made. They just control frequency, quantity, that kind of thing. Just like enhancers do. This is yet another fine-tuning knob on that control. Okay, so we go from start to stop. That is our direction of translation. So the AUG start codon is going to be a methionine. So the methionine is the first amino acid of every protein transcript. Now, there is a thing called post-translational processing. And I alluded to this briefly when I was talking about the disulfide bridges of the cysteines in the previous lecture. I mentioned one particular protein. Some cysteines bind to each other through a disulfide bridge, and then the polypeptide backbone is cleaved, and so you actually have two subunits of protein attached and fused to each other. That fusion is a post-translational modification because it is a modification to the polypeptide that happens after translation. It's a very, easily defined name. Breaking of the polypeptide bond at that point is a post-translational modification. The additions of phosphates or, there's all kinds of things that get added to protein R groups. You can have glycosylation, you can have fucilization, just so many different lipids and sugars and carbohydrates that can get added to the R groups of proteins as a way of modifying them and changing their behavior. Phosphates, and you can also cut off chunks of protein. Many proteins have the first 15 or so amino acids cut off deliberately. That's just part of how the protein becomes functional. And in those cases, their methionine is one of those amino acids cut off. But as the protein is getting translated, always the very first amino acid, as it first gets translated, is always methionine. You can't say that about any other amino acid in any protein. There is no definitive, the 17th amino acid is always. No. Now, you can say in a particular group of proteins, like in all of the DNA polymerases, this one amino acid is conserved throughout everything. Sure, but you can't say that for all proteins. The methionine, you can. The stop codon does not encode an amino acid. So, and we'll get into that in a few minutes. The stop codon does not encode an amino acid, therefore, You can't say the last amino acid of a protein is this thing, because it's not. So what happens when you've got this mRNA? Well, this big purple blob is a representation of our ribosome, and the ribosome clamps itself onto, or gets clamped onto, the mRNA and shuffles along 3 nucleotides at a time, 1 codon at a time, and basically attaches amino acids together with peptide bonds. They're binding the carboxy group at the carboxy terminal end of one amino acid to the amine group at the amino terminal end of the next amino acid. That is called a peptide bond. And as the ribosome moves along the mRNA from one codon to the next, from the start towards the stop, it just keeps adding on one more amino acid at each codon. And it uses tRNAs to do it. Loaded and unloaded tRNAs. That's the short version. That is, in a nutshell, how translation works. But of course, that doesn't tell you anything useful, and that's, not even quite the level of information that most high school courses go into. So let's get into a little bit more detail here. Translation has 3 phases, just like transcription did. Initiation, elongation, and not termination this time, but recycling. However, before we get to initiation, there's some things we have to do. We have to prepare some ingredients. And in this case, we need to prepare our amino acids and our tRNAs. What, pray tell, is a tRNA? I'm glad you asked. A tRNA is a non-coding RNA. That is, an RNA that does not get turned into protein. It does not encode protein, much like UTRs. However, tRNAs are not the UTRs of other genes. They are just RNAs that are made specifically for the point of being tRNA. For those who believe in the RNA world hypothesis, this is one of the major pieces of evidence used to suggest that before there were cells, before there was DNA, before there were proteins, there was RNA doing work, catalyzing reactions, carrying information. That RNA was life. So, what's the structure of a tRNA? What are we looking at here? Well, the images on the left and the right are essentially the same thing, but they provide slightly different information. So I included them both. They're from different textbooks. On the left image, the green areas are areas that form double-stranded RNA. They anneal to each other. And the way they do that is there's a stretch of bases in between them, 7 bases, 11 bases, what have you, and that stretch between them bends and folds and brings the 2 other sides together, and those sides form hydrogen bonding, and there you go. It forms a bunch of stems and loops. And the loops have some specifically conserved nucleotides, and there's some positions that are important and specifically important. So the 2 things I want to draw your attention to are the very bottom of each image where it says anticodon, and the ACC, or the CCA end, I should say, at the 3' terminus of the sequence. The CCA end is where an amino acid will get covalently attached to your nucleotide chain, your tRNA. And so it's just a fusion of an amine group to the OH at the end of the ribose sugar. The anticodon is 3 RNA nucleotides that are essentially template strand. They are template strand for a codon. Now, this is what tRNA would look like if we lived in a two-dimensional world. We do not live in a two-dimensional world. tRNA folds. So the image on the left is essentially a copy of the image, one of the images we just looked at. The one in the middle is a sort of better version of what things look like. And I hope you can follow the geometry of just that 45-degree angle twist at the bottom end of the yellow loop and at the joint between the purple and the light blue. It basically folds along that line, and the image on the right is a ribbon diagram of how the nucleotides actually look. So not only do the nucleotides bind, they actually form quasi-double helices, because that's what nucleotides do when they're double-stranded. So here's a better image, I consider, of the tRNA, because it points out a few other specific things. I mean, this one, it's not just showing the sugar phosphate backbone, it's actually showing the hydrogen bonded nucleotides, the bases in between. And what you notice, the anticodon at the bottom, just because of the three-dimensional structure of how this thing turns, twists, and bends, the bases of the three nucleotides of the anticodon are facing outward instead of inward. And that's incredibly important. That is the entire reason tRNA functions. Now, in the slide describing who the players were, I mentioned acyl tRNA synthetases. That's a class of proteins, and their whole job is to bind a tRNA, recognize the anticodon sequence, and based on that sequence, attach the correct amino acid to the 3' acceptor, that adenine at the 3' end. Now, how do they know how to read the anticodon, and how does that dictate which amino acid they bind? Well, that's essentially called the second genetic code, and it's not something I'm going to get into in this course. Needless to say that there's some specific interactions between the acyl tRNA synthetases and the tRNAs. And one thing that I'll mention is the DNA sequence encoding each of the 20 tRNAs. And remember, there's 20 tRNAs, one for each amino acid. The DNA sequence that encodes a tRNA is present multiple times in a genome. So the tRNA that is for the amino acid threonine, there might be 25 different copies of that DNA in a given genome. And it's weird, but even though the tRNAs are identical, which one of them gets transcribed into RNA has an effect on the cell. And it changes things in the cell, and some of them are only transcribed in certain tissues and at certain times. And that's an area of biology that still isn't very well understood. But they're not identical. They are different in their function. But I think the sequences themselves are identical. It's just when and how many are made. And there are differences in how they function as a result. If someone were to get into cellular biology as their career, this would be a fascinating subject to investigate. One of my former lab mates, Maria Barna, she's out west, I believe, at Berkeley. Her whole lab, her field of study, is the fact that the ribosomes themselves are different and expressed in different cells and different tissues at different times. And that has an effect on the function of the proteins. And that was something that she discovered like less than 10 years ago. And it has been a massive shift and overhaul in how we see ribosomal RNA translation. It's still very unclear how that makes a difference, but it is clear that it does make a difference. So this is kind of cutting edge stuff we're talking about here, even though a lot of this was initially worked out in the 60s and 70s. Okay, so that was our pre-initiation gathering together of ingredients. And now we're going to get into initiation. There are differences in initiation between prokaryotes and eukaryotes. I'm going to discuss broadly what these differences are, but I'm not going to get into tremendous detail about it. The important things to remember are, with prokaryotes, there is no nucleus. The chromosome is in the cytoplasm, and as a result, the ribosome and the transcriptional machinery can exist side by side. And so as the mRNA is getting spit out the back end of the RNA polymerase, as it is very initially first being transcribed, the ribosome can attach to it and start translating the front half of the gene before the back half of the gene is even done being transcribed. Now, remember I said there could be multiple different polymerases on an mRNA. Multiple different polymerases on DNA making their own mRNAs. There can also be multiple ribosomes on an mRNA making protein. So just from one gene on one chromosome in a bacterial cell, you could have multiple RNA polymerases making RNAs, and each of those RNAs can have multiple ribosomes on them all at the same time, all at once. It can kind of look like a fractal diagram with trees branching off of trees. In eukaryotes, the RNA is transcribed in the nucleus, and then it has to migrate out of the nucleus into the cytoplasm to reach the ribosomes, because the ribosomes are only in the cytoplasm. As a result, the structure of the mRNAs are different, just by sheer necessity. Eukaryotic mRNAs have to last long enough to get from the transcriptional machinery to the translational machinery. They have to not be degraded on their migration from inside the nucleus to the outside of the nucleus. That is why they have the 5' cap. That is why they have the poly A tail. In prokaryotes, the mRNA starts getting turned into protein immediately. It doesn't need a 5' cap because there is not a chance for it to get degraded. So, prokaryotic mRNAs really only last a couple of minutes. They, once they've been turned into protein, or once protein has been made from them, they basically just degrade and go away. They only exist for a couple of minutes at a time. In eukaryotes, the mRNA has to survive longer. And so it actually, because of the cap and the poly A tail, they can survive hours and in some few cases, a day or two. So the RNA structures are specifically different. Now, how the ribosomes work, how they function is the same, with minor differences, but how they load onto the mRNA is different. In eukaryotes, the ribosome assembles, because the ribosome, remember, is a complex of multiple proteins and RNAs, that assembles at the 5' cap. But prokaryotes, their mRNAs don't have 5' caps, so their ribosomes cannot assemble at a cap that doesn't exist. So what do? They assemble directly at the start codon. So there is a short sequence that is very highly conserved throughout prokaryotes. It is a string of purines, and I'll let you remind yourself of what nucleotides purines are. It is a string of purines, 8 bases, 5 prime of the start codon. And that conserved sequence is named after the 2 scientists who discovered it, Shine and Dalgarno. So the Shine-Dalgarno sequence is immediately upstream of the start codon, and that sequence is where the ribosome assembles. And because the ribosomes are bulky and large enough, it actually is on the AUG when it assembles on the Shine-Dalgarno sequence. In eukaryotes, as I said, the ribosome assembles at the 5' cap, but remember, from the 5' cap to the start codon, you've got that whole 5' untranslated region, that whole 5' UTR. So what does the ribosome do? It assembles at the cap, and then it just drives through the UTR. It just drives along the mRNA until it hits the Kozak consensus sequence. What the heck is a Kozak consensus sequence? Well, number one, it is only found in eukaryotes because prokaryotes don't need it. They've got the Shine-Dalgarno sequence. The Kozak sequence is named after the scientist who discovered it. I believe her first name was Marilyn Kozak. And that is a roughly 7-base sequence that includes the AUG. So the Shine-Dalgarno sequence in prokaryotes is upstream of the AUG. The Kozak sequence includes the AUG. It's about 7 bases long, or maybe 8 bases long, and the AUG is bases 5, 6, 7. And then there's one more base after it, and it's almost always a G. You don't have to memorize what the Kozak sequence is. It is not a specific 8-base long sequence. There's always an AUG here, there's almost always a G there, and then the first four nucleotides are, generally of this type of thing. And then some of the other main differences are the size of the ribosomes. Ribosomes are larger in eukaryotes. And I'll get into what these nonsensical equations mean in a second. And in eukaryotes, there are more auxiliary proteins required than there are in prokaryotes. Or at least, there's more of them doing different things. Those are our broad differences between prokaryotes and eukaryotes. Let's get into some specific information here, shall we? Alright, so I'm actually going to stop the lecture here for today, and we're going to continue with the rest of translation on Thursday. I'm going to finish off translation, and then I'm going to start gene regulation, probably with the lac operon. And if there's time at the end of that, I really want to tell you guys the story of the discovery of mRNA, because that's a fun story. So, for now, enjoy this, and feel free to ask questions. Always, if you don't understand something, ask questions. Read the textbook, do any problems that are in the back of the textbook, or in the end of the chapter, and watch the videos I linked to. That should be good for this lecture, and yeah.